Okay, so today I'm going to be talking a little bit about some of the work that my team has done over the last, since like about 2011, 2012, and how that's inspired some of the work I am doing and would like to do in my research career, and how that works in data integration. So at CHOP, we have been investing in biobanking, which has led to a significant framework to use the biobanking world and collect tissue. So I'm going to talk a little bit about how we drive EMR-based phenotypic research in pediatric brain tumor research specifically by leveraging this infrastructure. So as Jeff discussed, I've been here for about 11 years. I supervise a team. We engage in really deep dive, complex repository-based research projects. And usually there's a very complicated data integration need in the operations component and the scientific component. I guess I'm paraphrasing all of this. We've built and published on specific components of CHOP's Biorepository Research Center, the software used to collect patient data for the biorepository. Again, I'm a Drexel alum. And my research interest is serving up that data for discovery in rare diseases through analytics, modeling, and machine learning practices. My work, that some of it I'm going to share with you, comes from my dissertation, which was funded by a Coherence at Scale program, actually a program in the humanities from the Council of Library and Information Resources. So today, we're going to learn how longitudinal biobanking here at CHOP and the software resources that contribute to it contribute to the molecular, clinical, and tissue repositories, and in turn, biomedical research. We're going to understand some of the challenges in phenotyping in rare diseases from the medical record, and view an application and analysis pipeline from the medical record based on lessons learned from a very well-annotated biorepository here at CHOP. I'm going to be taking there's going to be two parts of this presentation. I've never really done them together. I'm going to talk about the biobanking and the software, but then I'm also going to transition into my research interest, which is computational phenotyping of pediatric brain tumors and other rare diseases from very sparse and noisy data. So I'm going to kind of try and put them together. This is the first time I'm going to do this. We'll see if it's successful. So two things to note of definition. The computational phenotype, you've probably heard other speakers, if you come to Informatics series or go to Informatics conferences, you hear about this computational phenotype or data-driven phenotype. This is finding patterns in biomedical data, usually clinical data, to find maybe a risk for some kind of output or variable. A good example, and probably one of the best and easiest example to understand, is find patients who might be at risk for some kind of bacterial infection in the hospital before it happens. So that's a data-driven phenotype. And then, of course, a biorepository in the sense of human tissue. This is something that collects, stores specimens from people and the data for research and clinical care. And some of the data would be things like a diagnosis, phenotype itself, genetic, proteomic, other omic information, imaging, outcomes, so on and so forth. So here at CHOP, back in 2012, our Clinical and Translational Science Award, along with the Center for Childhood Cancer Research and a big project called the Children's Brain Tumor Tissue Consortium, really stepped up their game in funding biobanking here at CHOP, and biobanking beyond just genome-wide association studies, but actually biobanking tissue and longitudinal tissue collection and data collection. And their repositories needed to do that over time. They needed to do it open-ended. We didn't know how many times kids would relapse from cancer. They needed to be able to add data later or before it happened that they actually collected it, and that's what I mean by asynchronous. And we had ongoing and real-time privacy needs for the families and patients participating in these projects. One of the first use cases we put together, and as I said, this mostly was funded in the cancer domain first, was the brain tumor use case. And we thought this was probably the most complicated we'd get, so as an information systems professional, I thought, let's think of what the most complicated way we're going to collect data is. And this is what we came up with. Today, I still use this famous stick figure. He or she's been around for a very long time. So in the brain tumor world, usually what it triggers the biobanking component of a study or consent to a biobanking study would be an initial diagnostic from surgery. So when there's actual craniotomy and they pull tissue out and then they look at it under a microscope. We then consent a patient, we get imaging, we have tests, and we collect a bunch of data. That patient goes on a treatment regimen, which of course is important to the data. We then need to go back and add the demographics, the medical history, family history, and things like that. And then we need to follow this patient, hopefully indefinitely. Unfortunately, in the high-risk brain tumor areas, this doesn't really happen. At the same time, we've collected some blood from the surgical event. We've collected tissue. We've collected cerebral spinal fluid. We've done some extractions of those items into molecular components that we can then run whole genome sequencing on or RNA sequencing and then do some analysis from there. The patient's life still goes on, and unfortunately, these diseases come back. So we have a recurrence, another surgery, more tissue might be collected. At that time, this could be a year later, and now we're collecting parental saliva, so we need to do that as well. New treatment plan, follow-up, this goes on and on and on. Unfortunately, with these diseases, in the high-grade world, this ends in death. So what our challenge is in the data world is to connect these things and make sure that they're ready for long-term use and research 5 years from now. So we need to know that this particular tissue came from this particular surgery. This particular tissue came from that particular surgery, and the other one came from the other one, because in cancer, the molecular makeup of the tumor changes. And then, of course, what we really want to answer is there anything that is in the molecular world that is making one kid surviving longer without a recurrence, or even surviving generally with the same treatment as another one? Why are they different? What's going on here? So those are the questions we're trying to answer in these biobanks. So when we first were given this problem by our Center for Childhood Cancer Research and our neuro-oncologists and brain tumor specialists to collect this data in this fashion, we first thought we have all these apps that do these things really well. We have one app that collects form data well. We have a medical record. We have a system in our biobank. We have all these different tools, but they're all different. We can't really get a one-size-fits-all solution. So I realized that, and talking to other colleagues, the solution is usually in our pockets. We work every day with the internet and interconnected web applications. And what's fortunate, just as an aside, when I show you this slide, I get to update it like every 2 years because we do all kinds of cool events here. So last year's Eagles Autism Challenge, I get to post on the internet that I'm collecting money, send that out to my friends and family. I raise money. I put pictures from last year of me and the Eagles cheerleaders. It's awesome, great time. I take a selfie of myself at this year's race. I make sure I cue up the music I like to hear in one ear. I am biking. I'm safe. And of course, I got caught by Chop's Instagram account, basically flirting with Carson Wentz to get a selfie of him, and I made him take the selfie. So they caught me doing this, and then I get messages from my friends telling me that I'm on Chop's Instagram being silly with Carson Wentz, which is fun, but I'm still biking. When I'm done, I get to share my pride and joy, my cycling stats with everybody on a different app. And then, of course, share on Instagram my newest Eagles cheerleader photo where I got photobombed by Swoop, which is cool, and, of course, the selfie with Carson Wentz. So this is really important. And when I get home, I can change my thermometer before I even get home. So what's going on here, and why can we do this here, and we can't do this for kids with maybe a molecular pathway in their brain tumor that we see in prostate cancer? This is the issue. And this is what we thought about when we were looking at our systems here at CHOP. We've got lab systems, data management systems, medical records, raw genomic data, query tools, all these things all over the place. We need to see them in different contexts. Why don't we connect them? So we used a lot of the same web-based tools to connect these. And we came up with this process where we, in effect, act as an honest broker to these different systems. And we do so in a secure way. And I'm not going to take too much of a deep dive into this. Again, I'm trying to do 2 presentations at once. This is another presentation in itself. And I owe a lot of this work to my colleagues in this room. But this is how we are doing biobanking today at CHOP. And this is not just how we do one-and-done biobanking. This is how we do biobanking that's going to be long-term and complicated. And I'm going to give you some other examples of this. So what are the results of this process of integrating applications? Best tool for the job, using web technologies. We now have almost 10,000 patients that have deep phenotypic data and a whole hell of a lot of biospecimens here at CHOP. This is just a representative of some of the specimens we collect here and across all of these different projects what we have. And these patients, almost 9,700 of them, have deep clinical data, whether it's from our medical record or it's been collected in forms specific to a domain. This goes on and on. We're now doing we have many studies. One of this is a good example too, where things can be complicated. Lifespan Brain Institute wants to study the use of melatonin and their kids' sleep patterns who may have a potential psychiatric disorder. Maybe they have signs of that. So they enroll these patients, they start following them, they give them a little actigraphy watch, they collect that data, they have a few clinic visits, they get a placebo or the melatonin. We collect some specimen blood at the time of the visit. They're doing biomarker studies. We're holding some of that in the biobank. They're coming in again and again, and we're collecting actigraphy data at specific time points, along with 3 biospecimen collection time points. And this one's very specific, but you can imagine the utility of this information being used by someone else later down the line. And of course, for the informatics stuff, we need to connect all these things. One of our newest projects is a birth defects biobank. We have a mother coming into our special delivery unit who is pregnant, and she has some clinical testing. The father of the baby comes in as well. We want to get his history. We want to get a maternal history, an OB history, a family history. We want to collect mom's blood. We want to collect the dad's blood. We want to do an ultrasound and get a preliminary diagnostic from that ultrasound. This baby gets delivered. We collect cord blood, placenta. Now we have a child here at CHOP. There's a NICU. The information's in the NICU. We collect blood from the NICU. Now this kid has surgery. We collect blood and tissue from the surgery. We need to connect all of these things. We also need to connect mom and baby. This ends up with a current biorepository that contains blood from all of these different family members. So now we have genomes on all these family members, and even placenta. The brownish gray bubbles are actually different component parts of the placenta, and that's how they're labeled there. It's hard to see it. But you can see how powerful this becomes very quickly. So what happens here now that we're doing this biobanking? It seems that longitudinal clinical data plus biospecimens plus molecular data equals scientific projects. And this couldn't be more true for the Children's Brain Tumor Tissue Consortium. And that is a multi-institutional brain tumor biobank that is where the data coordinating center and specimen coordinating center is here at CHOP. I've worked on this project since its inception. It's kind of my baby. I love the project. I love the idea of this project helping kids with these rare diseases where there's really just no treatment for them. If you look in the bottom right-hand corner of this infographics created by that consortium, there are over 50 scientific projects going on now based on these data points, this molecular data that is now available to this community. And what's in this data are information from the tissue. So we've got normal tissue, we have tumor, we have somatic DNA, we have tumor DNA, we have all of these different things. We of course have lots of analyses that other people have done, and we have the sequencing that is available for people. We have outcomes, some clinical data and treatment data of these patients, and of course we have the results of this being hopefully better diagnostics, predictive biomarkers, prognostic factors, and new drug targets that we can create based on this project. And if you've ever been to probably any conference where there's a high-level molecular discussion, you see that the cost of molecular sequencing and genomic sequencing has gone down. We all know that. It's going faster than Moore's law. But how about the phenotype? This is really where the issue is. Yes, it's becoming cheaper to know that there's a variant in someone, but it's still really expensive to know the phenotype, who that patient is, what is their description, what are their outcomes, what could happen to them, what will happen to them. So the analysis of phenotypes is lagging way behind our ability to do this on this industrial scale we're doing it now. So longitudinal clinical data in precision medicine is really important, and we have a lack of consistent methods to prepare and analyze that data. And why in brain tumor research is it so difficult? Because in clinical research, where these kids are usually found to have lots of data and longitudinal data, automation of the data capture varies. The human burden of data entry is incredible. And the operational logistics to make something future-proof, which I call it, the resources are expensive and unpredictable. There's lots of turnover in staff, and then sometimes projects just close and the clinical trial closes. So we don't really have the same data across the board for these kids. And of course, institutional record keeping varies from hospital to hospital, so that's even more difficult. The question is how does a group like the CBTTC do it? Well, they have human curated electronic forms. And this is across now I think 16 institutions. This is just a few of them. This is scalable for this type of project. We've seen it be scalable, but there's something here I think we could maybe do better with phenotypic analysis for these children. So where can we get this clinical data? We know we have clinical data research networks like PedsNet, which is based here at CHOP, which uses data from across many institutions, puts it together, and it's observational clinical data usually used in the more common diseases. Of course, the medical records at different institutions. We've got registries in rare diseases and specific diseases, insurance companies. I don't know. There's lots of ways to get this clinical data, but it is there. This is the thought that came to me when we were working on the third project for the Children's Brain Tumor Tissue Consortium. This is the third out of now 50. We were working with Seattle Children's Hospital, and we had a researcher there who was very interested in a specific high-grade glioma. At the time was called primitive neuroectodermal tumors, and they are now not called that anymore because of the molecular research that's gone into them. They're now subtyped into like 120 different types, which is pretty interesting in itself. But what she wanted to look at was the molecular underpinnings of the diagnosis, the disease location, where it shows up in the MRI, the treatment and survival for different kids, and what are the molecular components of that tumor versus other kids with the same tumor or different treatments, and come up with, in cancer, different survival measures for and pair that with the molecular data. But what she really needed was, if you remember the graphic from before, was all of this clinical data in between the molecular and the outcomes. So this is where I started thinking, because I was part of the team that actually had to pull the data together, so that was hard, and I realized it. So what I thought we needed to do was build a framework for demographic, clinical presentation, treatment, and outcomes from observational data sets. And we had two institutions to work with. We had Seattle Children's Hospital who were very much part of this project, and we have Children's Hospital of Philadelphia and many other participants. But we had physicians at both hospitals to work with. We had to use the noisy clinical data to represent all of this biomedical research and hopefully be able to reproduce this process in some way. As I mentioned before, there is a government project called PedsNet, which is part of PCORnet, which does host, I think now, 6 million open-ended pediatric observational data sets. This is all aggregated in a really nice and friendly data model. It's usually used in common diseases, but what I saw here is that we have two overlapping institutions in the Brain Tumor Project as well as in the in PedsNet, and that's Children's Hospital of Philadelphia and Seattle Children's Hospital. So the question is, I then made a request of PedsNet for all kids with high-grade gliomas within a certain time frame we had for these brain tumors, as well as the ones we may have biobanking, knowing full well that the kids actually are in both data sets. So I did receive a very large data set that included 4.3 million events over 16,000 patient years, meaning like that's how many, if we lined all the kids up, that's how many, that's how long these kids are in the hospital. So we've got a lot of data now. And the question is can we find a computational phenotype and limit the burden of human data entry on this? Of course, that was my first question, because I have it as the second question here, because I was one of the people who actually had to pull this data together. But there's also more to this. Can these large data sets provide new phenotypic research based on what we find in these records? We should use them for something. We do have them. So, of course, my question is there something to this overlap here? So we started with an exploratory data analysis of these patients, pulled out about 90, and these 90 represented potentially what looked like the patients in the Seattle third CBTTC cohort. So we pulled them out, and this is really the noisy data, what you would get for a patient, and all of those concepts on the left, and then over time, how many of those times those things happen. What I just also decided to do was to take kids out who had neurosurgical events, meaning that there was a suspected malignancy in the brain because they had a brain tumor, and usually they have to go into the brain to pull that out to find that histology. And then taking, I guess, the usual experience in dealing with data, and that is trying to categorize this stuff. You have this whole mess. What are you going to do with it? And that's categorize it. So start with ontology. Start with what we know. We know that SNOMED works in a polyhierarchical manner. So let's start to normalize some of the diagnoses we see across the problem list. So bring up the diagnosis. We don't know how someone's going to be diagnosed as Seattle versus CHOP. So we can start to bring them up, start to categorize them all as one thing if we need to. So SNOMED provided a little bit of that help. Also, we were able to use CPT codes because of billing. And billing, of course, drives everything. Money drives everything. So that was actually more of a help than the SNOMED codes, meaning that I could tell if someone had a surgery, if they had a radiologic diagnostic, if they had radiation therapy. All of these things definitely happened because we charged for it. So this was helpful to do too. So using lots of different matching patterns and so on and so forth, we could do this with those events as well as drugs. The NCI provides great dictionaries. They've been doing this for a long time on what's a chemotherapy, what's an immunotherapy, and other combinations that could be for cancer treatment. So I was able to pull those out with lots of word matching and things like that. So what came up from this categorization, just looking at time, was taking 2,000 unique events across maybe one patient and paring that down to 50, which is much more readable by a physician. So I was able to go back to a physician and get these patients all lined up for potential predictive analytic work. So from that really fuzzy, messy mess to something that is much more readable by a physician to something where I could then confirm with the physician that this is actually the case we're looking for. In the bottom, your bottom right, there's one patient, and those are the diagnoses in the problem list for this kid. And what you see are 4 different diagnoses of 3 medulloblastomas and 1 PNET. And PNET was what I was interested in, the third one down, primitive neuroectodermal tumor. And when I showed this to one of the neuro-oncologists, she said to me, actually, no, this is not a PNET, this is a medulloblastoma. In this year, or whatever the year was she was talking about, we categorized all recurrent medulloblastomas as PNET. That is no longer correct. So having a human look at this is very important. So getting the data prepared in a way that is ready for analysis to work with the sample is very important to make it translatable to a human. Again, people-focused data integration. It's again, hits me right in the head. So in summary, all of that, the methods to get the data where it needed to go was a derivation of a sample set, of an assumed sample set, evaluation of the sequential events and temporal data, find possible categorization, anything we could find. And in this case, it was SNOMED, CPT procedures, and the NCI had some really good resources on drugs and procedures and what we should be using in cancer. So categorize, reduce features, matching, and sometimes some just plain old logic to get where we need to go. And then evaluate those outliers like this one with subject matters, experts. So what are our results? Because of the temporal proximity of events in these kids, again, this is a very deadly disease, so we see it happening at a certain age, and usually we only see these kids surviving for a few years. And most of the kids in this group will be in that little time frame. So I'm able to use days, which is terrible, but it is the case. So what we can do is we can line up the temporal sequencing of this, and we can do different density distributions of these children's experiences here having a brain tumor and at Seattle Children's. So this density curve visualizes different events in those categories I showed you. And this reflects what I think came up very quickly to me, is that this reflects this situation that I showed you in the beginning when we were discovering our initial use case on cancer biobanking, that this activity right here equates to an initial diagnosis, a pathology report, imaging tests, comes out of all of those places. This part here is the follow-up and treatment. This part here is a recurrence, another surgery, actually a neurosurgery, imaging and tests. And of course the follow-up and treatment, unfortunately, I think these kids do pass away, so we don't see them just going home. And that's why we're doing this. And to just drive this home, this does actually, I pulled this record out, this does actually equate to, you can see at the bottom, this is actually in order first event to the top. So we see a spinal puncture, so for diagnostics, we see a chemotherapeutic kind of combination for this kid. We see imaging being done, and we see a lot of labs and monitoring of this kid through their process here. Now, what's great is you can take every single one of these 90 patients and line them up the same way and see what their activities looked like. So these patient-by-patient density grids could be pretty powerful. So what I found by doing this work is that, of course, we have these supervised brain tumor data sets, but where this could go would be potentially doing unsupervised feature sets in this brain tumor data set. And I am working on that with a physician here at CHOP. Who works in epilepsy. So we're both working together, and he has an epilepsy data set, I have a brain tumor data set, and we're trying to do this in an unsupervised fashion. So basically we're starting over. reevaluate the importance of this supervised versus unsupervised approach to EMR data, especially for me in rare brain tumor research, and then hopefully link that information that we find, these categorizations, this clustering, into actual biomedical research. Maybe the clustering that we're finding computationally could have correlations to the molecular findings we have. And then, of course, expand the disease base and get out to other areas of oncology or other biobanks that we're working with. Where this, and as Jeff explained, I'm very interested in operations. So like, what does this really mean for us here at the hospital and for research operations? It's one thing to talk about clustering. It's one thing to have pretty diagrams. It's another thing to make something work here at CHOP. And what originally started out with this project was, I want to automate some of this manual data abstraction. But these things actually may equal points in time where we need to collect the data, where us as an institution need to be focused on collecting biospecimens. We may not have all of this work set out already in other diseases. In other diseases, we may just have some subject matter experts that can go through this process and then say, okay, well, we know that this is an important time point. If you see some action in these categorizations, let's keep those blood samples. Let's keep that tissue sample. Let's keep these blood gases. We actually don't know, but maybe there's something to this. And that's something else I'd like to kind of look into here at CHOP in our biobanking strategies across the institution, and not just for these specific diseases. So in sum, phenotyping is expensive, but we can plan for it. Translational data plans are now seeming to be part of grant write-ups, and I think they should be. Honest brokering systematically has led to an unbelievable use just in the last couple of years of data here and research data here at CHOP. Rare diseases can't be studied in a vacuum. We need to share and we need to standardize. And then, of course, why this research was funded in the humanities was because there are humanities-driven groups that want to bring these problems out to other people than just physicians and physician researchers. We need computer scientists at the table. We need information scientists at the table. We need engineers at the table, and we need artists at the table. We need everyone at the table. So how do we make the data in a way that everyone can deal with it or look at it in a certain way? So with that, I open the room up for any questions. And these are I just wanted to leave you with this. When I was working on my dissertation and I proposed my thesis, I presented my committee with this IKEA instructions that reminded me of what it was like getting a PhD. And I still to this day think that this process was like IKEA instructions. In fact, I can actually put this whole entire process on top of these IKEA instructions. So if it sounded like IKEA instructions, I apologize, it was. So if you have any questions, please feel free to ask. Thank you for your attention and time.